Hi viewers, in this class we are going to discuss cell mediated immunity apart from the types of immunity. Under this topic, we are going to discuss the definition of immunity, types of immunity, definition to cell mediated immunity, cell mediated immunity responses, development of T cells, and the mechanism of the cell mediated immunity. Here, immunity is the ability of the organism to defend foreign antigens, microbes, and the pathogenic agents. Now, this is divided into two categories based on the involvement of the B cells, T lymphocytes, and other cells. If the B cells are antibody producing cells, if they are involved in the immunity, that is known as the humoral immunity because antibodies are secreted into the body fluids. These body fluids are called as the humors. Whereas, majorly the T lymphocytes, along with the T lymphocytes, besides the T lymphocytes, there may be the involvement of the macrophages and the natural killer cells in the cell mediated immunity. Cell mediated immunity is the immunity which is mediated by T lymphocytes, that is T helper cells and cytotoxic T lymphocytes. Besides that, there may be the involvement of the phagocytes and the natural killer cells. So, these are the B lymphocytes and they are producing plasma cells, memory cells. Plasma cell produce antibodies, that is the humoral immunity. Whereas T helper cells, cytotoxic T lymphocytes, and the subsets of T cells, they mediate the cell mediated immune responses. So here this is the humoral immunity. So main responders are the B lymphocytes. The cell mediated immunity, main responders are the T lymphocytes. And come to the cell mediated immunity. The term cell mediated immunity was originally coined to describe localized reactions to organisms, usually intracellular pathogens, mediated by lymphocytes and phagocytes. However, it is now used often in a more general sense for any response against organisms or tumors in which antibodies play a subordinate role. It is not possible to consider cell mediated immunity and antibody mediated responses entirely separate. Cells are involved in the initiation of antibody responses and antibody acts as an essential link in some cell mediated reactions. Moreover, no cell mediated response is likely to occur in the total absence of antibody, which can modify cellular responses in numerous ways. For example, formation of antigen antibody complexes during an immune response leads to the release of chemotactic complement fragments, which enhances accumulation of cells and local inflammation. Antigens would otherwise be targets for cytotoxic cells, may be modulated or stripped from cell membranes by antibodies. Antibody may also be involved in linking antigens to cells via the Fc receptors. Fc means fragment that is crystallizable, which is present in the heavy chain of the antibody, thus modulating the cell's responses. In the case of phagocytes and killer cells, antibodies can link them to their antigens. Cell-mediated immunity effective against both exogenous and the endogenous antigens, we should remember. Here, see, the T cells are mobilized when they are encounter a cell such as a dendritic cell or B cell that has digested an antigen and is displayed antigen fragments bound to its MHC molecules. So cytokines help the T cell maturation. The MHC antigen complex activates the T cell receptor and the T cell secretes the cytokines. And some T cells become helper cells and secrete some cytokines that attract fresh macrophages, neutrophils, other lymphocytes, and other cytokines direct the recruits once they arrive on the scene. Some cytokines spur the growth of more T cells. Some T cells become cytotoxic cells and track down cells infected with the viruses. Here, there are different kinds of cell mediated immunity responses. They are namely T cell independent cell mediated immune defense mechanism, T cell dependent cell mediated immunity or immune responses, cell mediated cytotoxicity, antibody independent cell mediated cytotoxicity, antibody dependent cell mediated cytotoxicity, lymphokine mediated activation of the macrophages, and the granuloma formation. These all are the side headings which we will discuss later on in the topic under the cell mediated immune responses, CMI responses. Let us take one by one the T cell independent cell mediated immune defense mechanisms. So, under this, we will explain the phagocytosis process. What happens in the phagocytosis? Here, as we know, there are different steps. Chemotaxis. What is the chemotaxis? Numerous microbial components will cause the chemotaxis of phagocytes towards the site of infection. Some, such as bacterial endotoxin, do this by activating the alternative pathway of the complement cascade, so releasing C5a and C3a. Microbial components like the formyl peptides are chemotactic and also directly stimulatory for phagocytes which have receptors for these common bacterial products, and thereby binding and uptake of the microorganisms. The binding of the phagocyte to the microorganism is facilitated if complement has been activated by the classical or alternative pathways. This leads to the deposition of the C3b on the organism, and the interaction between the CR3 receptor is an efficient way of triggering antimicrobial mechanisms in the phagocyte. Similarly, if antibody has bound to the pathogen, Fc receptors on the phagocyte may contribute to the uptake. Thereby, there is a cytokine release. In the initial stage of infection, the triggering of cytokine release from the macrophages play a vital role. Cytokines are peptide or glycopeptide mediators released from the appropriately stimulated cells. The first cytokine to be identified came from the lymphocytes and were termed lymphokines. The term cytokines now include interleukins, interferons, colony stimulating factors, tumor necrosis factors. The most important of the mediators released by the macrophage in response to the microbial components is tumor necrosis factor alpha. This is synergy with other mediators has several rapid protective effects. And thereby, here the ingestion of the microbe by the phagocyte takes place. This forms a vacuole, which is nothing but the phagosome, phagocytic vesicle. In order to distract this, there is a attachment of the lysosome to the phagosome, forms the phagolysosome, and the lysosome releases its enzymes into the phagosome. Thereby, the microbe will be partially digested, and the undigested or indigested material which is present in the residual body, that will be ejected out into the body fluids. So, this is about the phagocytosis. So, which is under the T cell independent mechanism. It never requires the T cells. And coming to the T cell dependent cell mediated responses. So, here we will discuss about the T helper cells. What is the regulatory role of the T helper cells? They direct fine specificity of the response. They determine which antigens and which epitopes are recognized. They are involved in the selection of the effector mechanisms to be directed against the selected target antigens. They aid the proliferation of the appropriate effector cell types. They enhance the functions of the phagocytes and other effector cells. And here we will see the role of the T helper cells in the selection of effector mechanisms directed against target antigens. The three most easily recognizable patterns of the effector mechanisms which can be selected are cytotoxic T cells, antibody plus mast cells and eosinophils, macrophages activation, delayed hypersensitivity. The decision is so important that activation of inappropriate effector mechanism can lead to enhanced susceptibility rather than protection. For example, in a model of influenza virus infection mechanisms, cytotoxic T cells protects whereas macrophage increase susceptibility. Antigen presenting cells present processed antigen to helper cells, that is the T helper cells, which are central to the development of the immune responses. These cells recognize particular epitopes and select those which act as the targets for the relevant effector functions. They then select and activate the effector cells. They can help B cells to make antibody and modulate the action of variety of other effector cells, including cytotoxic T cells, natural killer cells, macrophages, granulocytes, and antibody-dependent cytokines, cytotoxic cells. And both T and B cells may in turn be influenced by the T lymphocytes only. So here we will see the macrophage, which is the antigen presenting cells. These are the T helper cells. They in turn activate the other T lymphocytes and they may also stimulate the B cells. Here also, this is antigen presenting cell. There is interaction of the T helper cell, and due to interaction, there is a development of the T helper cell population and cytotoxic T lymphocytes, activation of these, and T memory cells. So these cytotoxic T lymphocytes, they may kill the target cell, and here the T helper cell interacts with the antigen presenting cell and secretion of the interleukins that may activate the natural killer cell, B lymphocyte, or cytotoxic T lymphocytes. So here in this process, the T helper cell plays a major role. So here activation of helper T cells we will observe. And next one is the cell mediated cytotoxicity, where there is active involvement of the cytotoxic T lymphocytes and the natural killer cells. The cell mediated cytotoxicity is the not only property of the cytotoxic T lymphocytes but also of certain other subpopulations of the lymphoid cells. The term covers different distinct phenomena. These cell types do not necessarily operate in the same way. Thus, different receptors can be involved in the binding of cytotoxic cells to the target and in the triggering of the killing pathways. There are several killing mechanisms. The main types of receptor ligand interactions involved are: number one, the specific antigens recognized by MHC restricted T cell receptors of T cytotoxic cells. Number two, determinants recognized by receptors on natural killer cells. Number three, antibody already bound to the antigen recognized by FC receptors of the killer cells. So here you can see the antigen presenting cell, CD8 cells. Here the mature cytotoxic T lymphocytes can kill the antigen perfectly. And there is interaction of the cytotoxic T lymphocytes with the cancerous cells. Antigen presentation done by the APC through the MHC class II molecules. These activated T lymphocyte release cytokines. These cytokines activated the population of the cytotoxic T lymphocytes. They bind with the cancer cell and kill the cancer cell in the programmed cell death, that is the apoptosis. Cytotoxic cell activation and the action. Here there is interaction between the antigen presenting cell and the cytotoxic T lymphocyte. These are the memory T cells and activate cytotoxic T lymphocytes. They kill the cancerous cell or infected cell. So the target cell dies off. So here also the same thing. And here the cytotoxic T lymphocytes not only interact with the natural killer cells but also natural killer T cells also involved. NKT means natural killer T cells. So these are the hematological malignant tumor cells. There is a switching of class 1 molecules and class 1 MSC molecules which activate the CD8 cells. In turn, natural killer cells activated, and here there is activation of the natural killer T cells. The killing is achieved. The killing of tumor cells achieved. Then antibody independent cell mediated cytotoxicity. Here there are different kinds of cells like the MHC restricted TC cells, MHC unrestricted TC cells, natural killer cells, and the lymphokine activated killer cells. LAK means lymphokine activated killer cells. Let us see the MHC restricted TC cells. As we know, the MHC restriction always the antigen presenting cells. They have in general MHC class 1 molecules are present on all the nucleated cells. They represent all these cells, all these antigens to that of the cytotoxic T lymphocytes. That is, there is a restriction in between MHC class 1 and the CD8 cells or cytotoxic T lymphocytes. Whereas the antigen presenting cells like the macrophages, dendritic cells, they have the MHC class 2 molecule through which the processed antigen will be presented to CD4 T cells. So after the presentation, after the exposure of this antigen to that of the immature T lymphocytes, T helper cells, they become mature T helper cells. That is, MHC restricted cytotoxic TC cells are subpopulations of small lymphocytes. The majority are the CD8 cells and recognize antigens in association with the class 1 MHC molecules. About 10% of the MHC restricted cytotoxic T cells are functionally distinct, being CD4 and MHC class 2 molecules are restricted. It is the CD4 or CD8 molecule which interact with the appropriate MHC molecule. It helps in cell-to-cell recognition. Antibodies to any of the cell surface molecules can inhibit killing. The most important role of cytotoxic T cells may be elimination of virus-infected cells. It therefore makes sense that cytotoxic T lymphocytes tend to recognize antigen presenting by MHC class 1 molecules, since these are expressed on extensively all nucleated cells. That should be remembered. Come to the MHC unrestricted cytotoxic T cells. They are NK cells present in spleen or peripheral blood. LAK cells, lymphokine activated killer cells activated by interleukin 2 and mixed population with non-specific killing activity. If we see the natural killer cells, NK cells mostly derived from the large granular lymphocytes, LGL. They comprise of about 5% of the human peripheral blood lymphoid cells. The majority of NK cells are CD3, CD16, CD56. Tissue distribution of NK cells differ from macrophages. Cloned NK cells have heterogeneous targets. Whereas LAK cells, lymphokine activated cells, this is the activity of the NK cells. Here, the lymphokine activated cell, there is increasing evidence that the enhanced cytotoxic activity of peripheral blood or spleen cell populations that have been pre-cultured with interleukin 2 is largely derived from the precursor cells which are indistinguishable from the natural killer cells. Thus, the lymphokine activated killer cells do not represent a separate lineage but rather the consequences of the activation. The difference in specificity may also be attributable to activation and to the changing contribution of other receptors. This type of cell is undergoing trials for the treatment of cancer in man. The patient's own T cells are simulated in vitro with IL-2 and then returned to the patient. And mixed population, here you can see the macrophage which is attached to the antigen, cytokines are released. The antigen presenting macrophages, they present the antigen to T cell, produce lymphokines. These lymphokines are activating the macrophage. These activated macrophages produce cytokines, antimicrobial function molecules, and the productions which are anti-tumor in function. And there are mixed population, stimulation of peripheral blood cells by autologous tumor cells, mixed population, lymphocyte culture, Phytohemagglutinin gives rise to many CD3 or CD8 cells which show typical class 1 restricted cytotoxicity. Some CD3 cells that express either alpha or beta or gamma or delta chains of T cell receptor and whose cytotoxic effects show little or no apparent MHC restriction. These are common when the phytohemagglutinin has been used as the stimulus in the case of mixed population. So these are the lymphokine activated killer cells which leads to the tumor regression. Coming to the antibody dependent cell mediated cytotoxicity, which is commonly called as the ADCC. Cells with cytotoxic potential which also possess FC receptors for immunoglobulin G may bind to and lyse the target cells coated with immunoglobulin G. Since some cytotoxic T cells and natural killer cells have such FC receptors, they can also perform this function. Observation of single cells have proved that both killer and natural killer activity can be properties of the some cells. The models most frequently studied involve antibodies to viral antigens on the membrane of the target cell membrane, determinants present on the tumors or particular MHC molecules, heptanes such as TNP conjugated onto the cell membrane, trinitrophenol, TNP means trinitrophenol, and membranes of avian nucleated erythrocytes. Myeloid cells expressing FC receptors can also show killer cell activity, but they probably use different killing mechanisms. The monocytes polymorphs may also be active against antibody coated tumor targets. Some myeloid cells, they are the monocytes and eosinophils, are certainly important effectors of the damage to antibody coated cystosomulae. In this reaction, the important antibody classes appear to be the anaphylactic ones, Ig in all species, IgG in mice, and IgG2A in rats. This raises the intriguing possibility that IgE acts by first triggering master cells to release eosinophil chemotactic factors and then binding the arriving eosinophils onto the target. This pattern of effector mechanisms is characteristically evoked when the helper T cell response is dominated by T helper 2 cells, since the IL4, IL5 reduced by these cells enhance the generation of eosinophils, mast cells, and the IgE. Then the NK cells killing the infected cell which are bound to. Then here if you observe the mechanism of the cell-mediated cytotoxicity, it appears that mechanisms involved in killing are similar whether TC cell, NK cell or killer cell are involved. The cells bind to the target first, and there is a calcium 2+ dependent phase in which the vesicle contents of the cytotoxic cells are discharged, and in the late phase when the target cell is killed. So there is the involvement of the perforin molecules and serine esterase enzyme and chondroitin sulfate A, these all are involved. And coming to the lymphokine mediated activation of the macrophages, here we will see the heterogeneity, calcitriol macrophage activation and macrophage regulation. Generally, macrophages act as a rapid protective mechanism which can respond before T cell mediated amplification has taken place. Then they take part in the initiation of T cell activation by processing and presenting the antigen. Finally, they are important in the effector phase of the cell mediated response, followed by T cell mediated activation as inflammatory or tumoricidal or microbicidal cells. Circulating monocytes possess the ability to kill some organisms. Much of the ability is lost if they are cultured in vitro, but exposure to lymphokines, particularly IFN gamma, interferon gamma, restores this ability and also activates additional killing pathways which normal monocytes do not express. The destruction of many intracellular parasites and of some tumor cells in vitro requires the lymphokine mediated activation of macrophages. Activation of macrophages involved an antigen specific pathway, but that the enhanced microbicidal activity was not specific and was known to be a property of lymphocytes and microbicidal activity a property of macrophages. If you observe the heterogeneity of the activated macrophages can express numerous different effector functions. The monocyte or macrophage series is very heterogeneous and cells taken from different sites differ such relevant characteristics as expression of class 2 MHC molecules and FC receptors, lymphokine responsiveness and production of peroxidase. The functions activated may depend not only on the macrophage but also on the precise blend of lymphokines and inflammatory stimuli to which it is exposed. Macrophages and their products are important in both the induction phase of inflammation and tissue recognition and repair. Macrophage effector functions may cause tissue damage as in hypersensitivity reactions. The activation of macrophage may require interactions of several lymphokines and microbial factors. Optimal release of tumor necrosis factor alpha from macrophages requires activation by TNF alpha, IFN alpha, followed by exposure to microbial components with cytokine triggering ability such as endotoxin. This may then provide enough TNF alpha to trigger the pathway 2. In the second way, IFN gamma activates the pathway which leads to production of nitric oxide, but TNF alpha is required in order to trigger it. And there is a role of calcitriol in the activation of human macrophages. When human macrophages are exposed to IFN gamma, they express a 1-hydroxylase. This enzyme enables them to convert inactive circulating 25-hydroxy calciferol into the active metabolite 1,25-dihydroxy cholecalciferol, that is nothing but the vitamin D3 or calcitriol. Macrophages have receptors for this derivative and it exerts additional activating effects on these cells and perhaps some negative feedback on the lymphocytes. And negative feedback of the negative regulation of the macrophage and effector functions. There is also evidence that activated macrophages can be deactivated. Prostaglandin E may have this effect and some effector mechanisms are steroid sensitive. Recently, macrophage deactivating factor MDF has been purified from a tumor cell supernatant. This factor blocks activation by IFN gamma of increased capacity of production of ROI and the nitric oxide, that is ROI means reactive oxygen intermediates. And coming to the granuloma formation, this is the last step in the cell-mediated immunity, granuloma formation. Sometimes the cell-mediated response fails to eliminate an infecting organism rapidly, or antigenic material cannot be eliminated because it is resistant to degradation or derived from self-components sometimes. In such cases, T cell continue to accumulate and release lymphokines. This leads to the granuloma formation. Granulomas are characteristic of infection with organisms which live at least partly intracellular, like the Mycobacterium tuberculosis, Mycobacterium leprae, Leishmania, Listeria, monocytogenes. And these all are the organisms which are large and persistent, like the Schistosoma ova. Granuloma characteristically contain macrophage derived cell types, somewhat obscure in function, including epithelioid cells and multinucleated joint cells. Their morphology suggests secretory rather than phagocytic roles, and they are thought to result from the different kinds of cells. like the macrophages or lymphokines. A granuloma can be produced experimentally using soluble antigen conjugated covalently onto an insoluble particle such as a saffrose bead. Antigen coated beads will evoke T cell dependent granuloma in appropriately immunized mice, whereas control beads will not form the granuloma. Analysis of T cells in granulomatous foci indicate that CD4 cells are located at the center and CD8 cells around the periphery, suggesting that CD4 helper cells are prime importance in inducing the accumulation and activation of other lymphocytes and macrophages. In mice, T cell dependent granuloma can be evoked by cystosoma ova, but they eventually wane under the influence of suppressor T cells. And coming to the mechanism of cell mediated immunity. Here, the mechanism of the T cell mediated immunity includes when antigen presenting cell process the exogenous antigen presenting cells APC process exogenous antigens, whereas the ASC altered self cell process endogenous antigen and present them to different kinds of lymphocytes. The processed antigen fragments are displayed on their APC and ASC antigen presenting cells and the altered self cells. So the processed antigens are displayed on the surface of these antigen presenting cells and the altered self cell membranes. They are recognized by the T cell. These all are recognized by the T cell. Binding of T cells to that of APCs and the ASCs cause the production of activated T cells and the production of memory cells. The activated T helper cells release or secrete the various types of interleukins which transform activated T cells into the effector cytotoxic T cells. Otherwise, the CTL, cytotoxic T lymphocytes, are killer cells. They attach to the infected cells and release certain enzymes called perforins and the granzymes. Perforins form pores in the cell membrane of the infected cell. Then granzymes enter the infected cells through the perforations and activate certain proteins like the caspases, which help in the destruction of the infected cells in the process of apoptosis, program cell death. The natural killer cells are similar in their action to cytotoxic T lymphocytes. However, natural killer cells destroy the infected cells in an antibody independent manner, whereas the CTLs, cytotoxic T lymphocytes, destroy the infected cells in an antibody dependent manner. So this should be remembered. This is all the mechanism of the T cell mediated immunity or cell mediated immunity. Here, in the first step, there is antigen presentation and T helper cell activation, cytotoxic T lymphocyte cell activation, and cell destruction, otherwise the destruction of the infected cell achieved. This is all about the cell mediated immunity and the steps involved in the cell mediated immunity. Thank you one and all.